that's great. Hello. We just have a yes. Morning. Yes, yeah, please let us know. Yeah, we're gonna try to use the whiteboard. Uh, so they will be able to see the whiteboard, right? People who
Whiteboard.
are this guy, the whiteboard.
A projector? Or are you using slides?
We will project some slides, and then I want to take it away and then use the markers and write on the board. That will be okay for.
It should be.
Okay. We'll see, I guess.
Which slides are you going to?
Matthias, I have everything
Go ahead.
here.
I just want to have the document open.
Oh, sure.
I didn't have questions.
That's a good question. They might be. If not, I can run upstairs and get some. Let's see if I can put this up.
Well, what would be nice at the beginning that we quickly go through the room and people tell who they are and what they do.
Not this one.
I'm adapted if you need an adapter.
Thank you. I should be able to figure this out. Supposedly at the Broad we've solved this problem in every room, but it should be. Hey, Sam. Morning.
Chloe, you went to get us some markers.
We did. Oh, great. I think that the idea was. And before that, we will just present a framework. And I just want to introduce you.
You guys are
Good.
when you're presenting
Yes.
here, you can use the podium.
Great. And then we can use the other mics
Walking around, or if anyone's going to be speaking, they have to pass these around
for the people to hear. Yeah.
Make sure at all times someone can
Say that again, please.
anytime you're speaking, just make sure you have a microphone.
And the mic is not going to the speakers here, but it's going out with the web.
It is going through the house in here, but most importantly, for the live stream.
But you will not hear yourself via the speakers in the room, so it looks like the mic is dead, but it isn't. That was a bottleness.
No, you'll hear yourself.
Oh, great. Yeah, that's good.
So, do you want me to read these?
Am I?
Any specific shares? Just carry them. Okay, there's the on-off switch.
Are we supposed to be projecting? Great, thanks so much. I should be. Oh, just right here. Awesome. Yeah, I'm sorry.
So maybe we could start. We're having some trouble with the slides, but I think maybe we just can get started. Good morning. I'm Anna Greca. I wanted to welcome everyone to the Kidney HCA session. I'm really excited to be here and learn from all of you, have the discussions around how we're going to make this happen. I want to introduce my co-hosts, Jimmy, Sam, and Matthias, who I'm sure will also tell you a little bit more about themselves and how they're involved in this project. I also wanted to, I was asked, and I think it's important to remind all of us that we are live streaming, and so all of this information that we're discussing is currently actively being live streamed, so that you're aware of that. As we try to get our slides up, we thought that maybe since this is not a large room here, I mean the room is large but not too many people, maybe we could all introduce ourselves. And actually we hope to use the whiteboard just like the Immune group did yesterday. We thought that was a very good way to organize things. Therefore, maybe one idea would be that we go around and you tell us who you are, and then maybe if you feel like telling us what projects are you already involved in, or what do you see in your world or in your realm as a project that is of interest to you in relation to kidney human atlas type of work. So maybe, yeah, we can start anywhere, please.
So, I'm Matthias Gratzler, University of Michigan, nephrology and computational biology. I'm involved with a kidney precision medicine project who is building a kidney disease tissue atlas. And we have had Aviv on our advisory board and at our kickoff meeting, who really, I think, give us an opportunity to align the activities here. And we have Minnie and Yul from the KPMP group here in the room as well. And looking forward to an exciting brainstorming session to get the Kidney Atlas going.
Hi, I'm Jody Hirschman. I work here at the Broad for the data coordination platform of the Human Cell Atlas.
Hi, I'm Laura Clark. I also work for the data coordination platform. I'm particularly interested in the metadata you guys need to collect. I obviously am aware the kidney is highly structured, and there are two of them, which makes life more interesting. So.
Hi, I'm Kevin Osborne. I'm in the data coordination platform section here, as you can see. I'm a PM for the data store portion of the data coordination platform, and I'm just here to learn.
Hi, I'm Wolfgang Ehnert. I'm here actually for the Standard Technology Working Group, which is one of those working groups that tries to think about how to implement standards and benchmarks. And originally, I'm an evolutionary biologist and did a bunch of RNA-seq comparisons.
Hi, I'm Ben Humphreys from Washington University in St. Louis, and we are performing single nucleus RNA-seq on healthy adult kidney and kidney transplant biopsies.
Hi, I'm Minnie Sorval, a transplant nephrologist, actually working with Matthias and other groups at the Kidney Precision Medicine Project, and also working with Jimmy at UCSF, actually doing a single-cell RNA-seq using multiplex technologies and demoxylate.
Hi, I'm Yoel Belize at the University of Michigan. I'm a pathologist and a member of the Kidney Precision Medicine Project, working on the central hub and the data atlas. As background, I'm kind of an accidental tourist in the pathology space. I'm actually an electrical engineer and machine vision scientist with imaging interests in content-based image retrieval.
Hi everyone, I'm Aishwarya Subramanian. I work here with Anna Greca and Aviv Regev, involved in single-cell RNA-seq analysis of kidney and also the HCA kidney efforts. I'm also your scribe for the day.
Hi, I'm Katrina Gold. I'm from the Wellcome Trust in London, so I'm a funder, and we are funding a kind of UK mini consortium which includes kidney atlasing components.
Hi, I'm Catherine Potensky from Nature Genetics. I'm an editor, and I think we're, from the genetics perspective, very interested in how the Human Cell Atlas and cell profiling can inform kidney, disease SNPs, and I think that's particularly true for the kidney, so I'm excited to be here.
Hi, I'm Aviv Regev from here at the Broad and from MIT and from HCA.
Hi, Asaf from Dana-Farber Cancer Institute. We have a collection of renal cell carcinomas, working with Aviv and her team on human cell atlas and tumor cell atlas.
Joshua Levin at the Broad. I'm on the STWG that was mentioned earlier, and I'm also involved in benchmarking the performance of single-cell RNA-seq methods.
Jonah Kuhl from the Chan Zuckerberg Initiative.
I'm Jeremy Miller from the Allen Institute for Brain Science. I don't know anything about the kidney, but I'm hoping some of the things we learn with single nucleus RNA-seq will apply to multiple organs.
Omar Bayraktar from the Sanger Institute. I work on the brain, but I focus on large-scale in situ spatial transcriptomic applications. And at Sanger, we'll be supporting Sam with some of his ambitions to reconstruct kidney cell types and tumors at the multiplex RNA and DNA level in situ.
Hi, I'm Partha Majumdar from India. We've started doing single nucleus sequencing, and we are looking forward to engaging ourselves in single cell sequencing in the oral epithelium.
Michael Reich from the lab of Jill Messerov at UCSD. We're creating a notebook-based portal to make the data that's deposited into the HCA accessible to researchers at all levels of computational sophistication.
I'm Paul Hoover. My primary appointment is through Brigham and Women's Hospital. I'm a rheumatologist by training with a special interest in kidney autoimmunity. I'm also a member of Neerha Cohen's group and part of the Kidney Precision Medicine program, work with Matthias, and then the Accelerating Medicines program that works on lupus nephritis in particular. We do single cell sequencing and try to do imaging as well.
Paul Robson from the Jackson Laboratory at Genomic Medicine in Connecticut. We do a lot of single cell sequencing as well as imaging mass cytometry. We've done a bit of work in the human kidney, but also we're very interested in the bladder and the transition from the kidney to the bladder down the ureter.
Jimmy Yee, UCSF. I'm a geneticist by training, and I work mostly in immune cells, but I've been doing and helping many with some multiplex single-cell nuclear sequencing, and I'm happy to sort of talk to you all about what I think are the ultimate and best experimental design strategies.
Hi, my name is Sam. I'm from the Wellcome Sanger Institute. I'm a pediatrician, and my main interest in the kidney is about tumors, really.
And I'm Anna Greca. I'm here at the Broad working with Aviv Regev on all things kidney, and with Ashwarya, who's in our group, and she's fantastic as a computational biologist. And I'm here to learn from all of you. And so I think our slides are not working, so we're going to just, Matthias, you can probably do it without slides, right? We will try. I think we should put up the... Yeah, exactly.
As there are multiple activities in the renal field already moving forward, maybe we can give a quick update what's happening on which fronts. And as Yul and Mini already have introduced, the NIDDK has provided significant programmatic efforts to build a kidney tissue atlas of disease states. Is this not working? No, it's better. Okay, so the philosophy there is that kidney is a very heterogeneous organ, and kidney diseases currently are defined in descriptive and not in molecular terms. And there's a clear need to fill that gap, to come up with molecular mechanisms which subsequently can be targeted by therapies and diagnostics developed for prognostic and predictive disease modeling. And with the heterogeneity of the kidney, there's a significant opportunity to leverage that using the single-cell technologies. And the kidney has two unique features which make that amendable from a disease perspective, and KPMP is disease-focused. That obviously for routine diagnostics we obtain renal biopsies. This is done for intrinsic glomerular diseases prospectively on every patient's undergoing workup, and there are efforts in the field to establish comprehensive atlases for intrinsic glomerular diseases, a rare disease network, which I'm leading, the Neptune network, has assembled a cohort of over 700 patients where we have kidney biopsy tissue profiled and available. So that's one trajectory which is moving forward. KPMP is focusing on the common diseases of the kidney who are coming with a higher societal burden, acute kidney injury, and chronic kidney disease, diabetic kidney disease. These people are usually not biopsied, and tissue is not accessible for analysis easily. And KPMP has developed a protocol to prospectively ascertain kidney biopsy tissue from patients who volunteer for a research biopsy. Meaning the tissue is procured in a defined, standardized settings with an opportunity to then process the biopsy course as comprehensively as possible. And it would have been nice to have a slide up to show the flow diagrams where the routine clinical biopsy cores, these are 16 gauge needle cores, they are usually 12 to 14 millimeters in length, processed on one hand, formalin fixed paraffin embedded, using the routine processing, providing routine diagnostic information back, but then also having an FFPE block available for future molecular analysis, biopsy core in OCT for analysis of frozen tissue, and then a biopsy core harvested in hypothermosol or snap frozen for the single cell technologies and spatial metabolomics. KPMP has 6 recruitment sites and 5 tissue interrogation sites. It's a distributed network to acquire tissue out of the broad spectrum of the society and then use a panel of currently developing technologies by the tissue interrogation site. Critically, it has a significant opportunity pool available to really leverage emerging technologies towards the biopsies material generated so that these biopsies are optimally processed to give comprehensive spatial and molecular information moving forward with a goal to build a tissue disease atlas, as Yul mentioned. This is a main feature of the community outreach, and KPMP is in the middle of getting a comprehensive use case capture of the needs of the community at large for the utility of such an atlas, for starting with the patients who donate tissue of their own body at significant personal risk for the advance of science. And we clearly will have a focus to serve them with the atlas. They have been embedded in the activity from the get-go, a part of the steering committee, providing the informed consent and the protocol with us. Then clearly scientists like those of us in this room, but then also clinician scientists in helping to help to generate information meaningful for clinical care, and researchers from the public and private sector. And there is a feeling that we need to have an adequate reference study in place to contrast the disease states, and that's, I think, where the synergy, obviously, with HCA become very obvious, and looking forward to see how that goes. And I certainly will bring what we discussed back to the KPMP group to see where we can align. People on the ground, Becky Steck, our project manager of the Central Kidney Tissue Atlas and Biorepository, and Tim Tickle are closely aligned. So from the backbone, hopefully things will be being matching together easily as we go forward.
Yeah, thank you, Matthias. I think we are sort of one step ahead in some ways in kidney, in the sense that, the NIDDK had the foresight to assemble this KPMP consortium and looking at diseased tissue, but the task is still at hand to really create a comprehensive atlas of what normal kidney looks like. And as was mentioned, it's a complex organ, highly structured. Disease affects different parts of the kidney in differential ways, and so to really try to define what's normal across lifespan, sex, race, in the most comprehensive way possible will probably allow us to really understand disease at the single cell level as well. And so with that in mind, I think we thought that we would actually use the whiteboard to sort of brainstorm about kind of specific areas that we thought would be important. And so Jimmy has kindly agreed to be our scribe on the whiteboard. But we thought we would split it up into experimental, which would involve a lot of the modalities that we might use for atlasing, which we can discuss. Also things about tissue procurement and study design. So those are the things that would fall under that category, and we wanted to sort of see what your thoughts would be on that. Computational is the next category, so we obviously want to be able to integrate data, see how we might be able to integrate other data or metadata with our initial atlasing efforts. And finally, of course, it's important to also consider validation and how that would occur in human samples, or whether there's other modalities that could be used for that. And so we just wanted to open it up and maybe have some microphones available around the room, because we always have to be on the mic in order for us to be heard in live streaming. But maybe we will tackle the first category first and really talk about you know, kind of experimental aspects of how we go about doing single cell in the kidney. This has been discussed, at least in the HCA sessions that I have been in, meetings that I have been a part of in the past. And so we do have some ideas about what works, and I think there's some experience in the room, but maybe we could think about, you know, frozen biopsy material versus cells, and how we would go about obtaining this tissue, maybe with the criterion across lifespan, race, sex, so trying to, you know, cover the globe, cover the entire human lifespan, including, you know, fetal kidney tissue potentially. And, you know, I know Sam has had some ideas about that, but also opening it up to anyone else who has ideas about tissue procurement, SOPs, sharing experience. Do you want to speak a little bit to that?
Perhaps we could start by discussing what doesn't work. That might be. So in our experience, the surgical resections are the best way to get data because the samples are relatively fresh. So initially I thought when a kidney is taken out, it's just taken out, but of course there's also an ischemia time of about 1 hour. So the way the surgeons do it, they go to the kidney, then they tie off the blood vessels supplying the kidney. So there's going to be at least 1 hour of ischemia, irrespective of what happens thereafter. And then you get your kidney, you can take your samples and process them straight away. And with that pipeline, we sort of managed to get from kidney to a single cell solution probably within sort of 4 hours. And I think that's quite realistic. The declined transplants, and this is quite interesting. So these are transplants that haven't been declined because of poor quality. They've largely been declined for logistical reasons, or there's a donor-recipient mismatch, or some other reason that the surgeon who receives the kidney didn't feel it was appropriate to implant. So although they're good enough for being a kidney in a human being, they're not necessarily good enough for single-cell RNA sequencing. And the data we get from declined transplants, that data are generally worse than what we get from fresh resection specimens. And then with nuclear, so we recently started doing some nuclear RNA sequencing on kidneys, and that actually looks surprisingly promising. And I do wonder whether it does give a more faithful representation of the tissue, because my main angst is that with fresh tissue, we've got all sorts of dissociation artifacts and biases in there. Aviv.
Sam, can you just confirm that the single nuclei works fine with the transplant? Is that what you just said?
Not with the transplant, just from fresh frozen biopsies.
I can make a comment on the nuclei. The first is that I would say that single nucleus RNA. You should actually say single nucleus RNA-seq if you say single cell RNA-seq. That's a pet peeve. But the single nucleus RNA-seq works well with the appropriate optimization on a diverse set of tissues, meaning the original protocols that we've developed for brain work really great for brain, and you need to do some changes in order to make them work for other tissue types, and this can vary from tissue type to tissue type. That's not so surprising, but I think those who have been using it in their labs have actually done all sorts of optimizations, likely compared to the original one. The original one is really good for neurons, by the way. The second point has to do with the question on dissociation artifacts. Dissociation does impact cells differently, and it leads to different viabilities and capture, and so there is a real bias that it can introduce. We originally thought that single nucleus RNA-seq would be free of these biases. I would say yes, it's free of dissociation biases. It can have its own biases. So not all nuclei are created equal either, and the process of isolating nuclei relies on the differential between what you do to the membrane, what you do to the envelope. Different cells are different. And so I think one has to be extraordinarily cautious about saying that something does not have biases, but it would have different biases. And so when you have the opportunity of take specimens and do both a fresh analysis and a nucleus analysis, if those are relevant in the kidney domain. I would strongly encourage doing it for a subset of samples to get an idea about representational biases, so that you would be able to compare one to the other and know this is good for this is good for that. You sometimes do that by covering the bases. Then there are additional approaches using spatial analysis to really convince yourself whether you're missing something that both cannot capture.
Ben, do you? Okay.
This is where actually in KPMP, in a pilot study, we used tumor nephrectomy samples, and Paul was part of that pilot to compare how the different protocols perform and what are the different blind spots are. And if you have time, Paul can comment, and I think, Ben, you spent a lot of time on that as well.
Yeah, so we had a pilot project through CZI whose goal was to compare different platforms head-to-head. And so we sort of came up with 4 conclusions at the end. And so these were samples that were processed equally, and one single-cell drop-seq, and the other single-nucleus dronk-seq, but we use 10x and it works just the same. And so the conclusions were interesting. In the single-cell dissociation protocol, we procured about 0.15% podocytes, but in the single nucleus, we had 2.5%, so 20-fold more, speaking to this dissociation bias issue. Of course, there were no dissociation-induced transcriptional stress artifacts with the nuclear protocol because it's all done on ice. But there's dramatic, as we all know, immediate early gene responses from single-cell dissociation that occurs at 37 if it's anything more than 10 minutes or so. The third conclusion was that it works very well for fibrotic tissue, and as Matthias was saying, KPMP is interested in disease, and CKD kidneys, chronic kidney disease kidneys, are very fibrotic and very difficult to dissociate, particularly in the interstitium. And so, again, I think that's a problem with single-cell protocols. And then the final conclusion was a big surprise to us, and that is that we achieved actually superior unique gene detection per nucleus and UMI counts compared to cells, even though it's much less RNA. And this was a big surprise.
So that's actually something we knew all the way back from the neurons, where this started was a tissue that you can dissociate, right? That's why we went there. And we believe that you get superior RNA detection, partly because the cells are so not unhappy, right, as they are in the dissociation process. So that's one piece. The other piece that I forgot to mention before, and it's actually relevant to something also Minnie and Jimmy said, is that nuclei you obtain from frozen samples. That means that you can accrue samples over time and then do some lovely experimental design. You cannot do that with fresh dissociation. And so you can control for batches, you can multiplex, you can do a lot of things that you just can't do with fresh samples because fresh samples are streaming mode. And to us, from an Atlas perspective, that might end up being one of their biggest benefits, that you would make one prefer them even in cases where you can dissociate.
Perhaps, one more thing. So Aviv is of course right that one needs to have that head-to-head comparison of nucleus versus fresh. But the issue here is we are comparing one bias to another bias, and the question really is, what is the gold standard? And we need to compare against the gold standard. And I wonder whether the gold standard in this context could be bulk RNA sequencing, because extracting RNA from either fresh or frozen tissue in bulk probably introduces, compared to the other ways of doing this, higher artifacts, and then have some sort of comparison of deconvolution or summing up of single-cell data to get a sense of, have we, first of all, captured all cells, and have we captured them in the correct relations, in the correct proportions? And then the other thing is, have we introduced new transcripts that are actually not present in the bulk, which could represent the artifact?
I would say from experience from the KPMP and the AMP, I think there's a few key points. Number one is the ability to collect the tissue and where that will be processed. And so for both of these projects, both for the KPMP and the AMP, and I imagine for the HCA, that whatever it is that you're going to have to freeze the tissue. And so that introduces its own artifacts. And you would basically, in order to reduce any kind of bias, you want everything processed at a centralized location. So that in and of itself presents some challenges. The second thing I would say is that we spent a lot of time actually trying to optimize different SOPs and dissociations. And we also tried to we also spent a lot of time thinking about how we would validate these. So we've systematically tested different dissociation enzymes, different conditions, what works best, and we focused mostly on single-cell RNA-seq. To validate these in both consortia, especially the KPMP, what we focused on is comparing single-cell RNA-seq to nuke-seq from the same tissue, and then trying to go back in and doing some type of image analysis to actually have different modalities to go in and validate these different cell types based on discriminatory gene sets that may overlap. This is obviously a very challenging component of it, but I think it's going to really be the key in order to actually validate these cell types. I think the biggest issue with a lot of our single-cell RNA-seq and nuke-seq is that we tend to, at least at the local environment I'm in, we tend to really place a lot of weight on these, but I would advocate that we need to have different modalities of validating these different cell types.
Just because Jimmy's actually, I think, writing this on the board, I wanted to come back to what Sam had said about transplant samples not being okay for single cell. So I think the standard workflow, yes, makes it very difficult to actually obviate that kind of time gap, and that time gap is actually quite critical, just like you said. But I just wanted to say, I mean, we have a very, very busy transplant program. We do about 400 kidneys, and we have a setup where we've finally been able to go into the operating room with our coordinators, and we actually collect the sample at source. That's incredibly difficult, I think, for people to do standardly, but I think if you can do it at that point, and then we've optimized our dissociation protocols where we're getting actually very good data, and we have some of that at UCSF, which we're not showing data today, but I think we can do it that way. So I just wanted to correct it because I think it says here that we can't, so just for that reason. The other thing is, I wanted to actually ask Aviv, because at one point we had talked about potentially doing nuke-seq of paraffin. Is that still a possibility? Because that would actually, of course, be wonderful.
I think one comment to one unique feature in the transplant setting is that many transplant programs, I think also Minisua, is performing protocol biopsies from living donor kidneys. And that, in our experiences using bulk studies over the last couple of decades, is this tissue which has a most pristine environment. And there are opportunities, as these are done for routine clinical care, to add an extra core to come up with material which is very limited in quantity, will be cortical kidney tissue only, as it will not be going into the medulla, but can serve as a reference point of studies which are coming from tissues which come with more constraints attached to them.
Yeah, I think we've got a slightly sort of semantic issue here. So I'm not talking about transplants from sort of live donors. I'm talking about declined transplants that have been hanging around for 12, 14, 18 hours in the system and being shipped around the country.
Actually, in the immunology session yesterday, one discussion point was, do we want purposefully include maximally activated tissue for the studies? Because it shows you what the organ can do at extremis and might indicate states which are more hidden and less frequent in the healthy state. And so deceased donor kidneys probably are as close as you can get an organ at extremis as possible.
Yeah, I mean, that sort of stuff lends itself to the perfusion rig sort of experiments where you take a kidney, put it on a perfusion rig, and then insult it in all sorts of various ways, and then try to read out what that insult does. I'd agree with that.
So this is another feature which I think is critical, that with the perfusion rig, there's an opportunity to really manipulate and functionally evaluate some of the conclusions which the descriptive categorization of the single cell analysis might bring to us, and that might be for the validation part later on.
So as I hear about some of these sort of challenges, are you guys making your data available out there, especially for these comparisons where, you know, you can probably write a paper, but I think it's much more helpful if you get these types of data out there as soon as possible so that people can look at it?
So we showed our comparison of cells versus nuclei at the ASN meeting just last week, and
it was even on a slide yesterday from many of the groups here.
Yeah. Yes. So, and I just wanted to say that from our experience, just to clarify that, you know, it's the discarded transplant kidneys that seem to be, you know, the worst kind of thing that you could get. But for freshly collected, like either tumor nephrectomies, or if you could sort of get a biopsy out of a kidney that's about to be transplanted, that's actually high quality tissue. So just to make sure that that's clear also for our audience on live streaming. And our group has figured out that the time limit is 1 hour in our hands. So it has to make it from the operating room to dissociation within 1 hour for the quality to be best. And so that's what we strive to do. And as Aviv said, this is sort of as they come in, whenever there's a procedure that someone is dedicated to go to the operating room and grab the tissue and run it over here for dissociation. And so when we come to study design, and I'm also mindful of the time, I'm giving each of the sections enough time for discussion, we should probably get to that, is how do we actually achieve study design that is computationally suitable and that addresses all of these other concerns about the quality of the tissue and therefore the modality that one might use for the single cell analysis. But I think you've been trying to say...
I'm actually trying to answer a question I was asked first from there, and then I think Mini asked the second one. So for the first one, I'll be very brief. We actually go in the white paper through a series of suggested strategies for how do you know that you got all the cells you were supposed to get. Like, what could you do by comparing two different methods? What could you do by comparing to spatial data? And so on. So I think it would be useful for this community, which I think was less yet on the kind of involved in the discussions that were now more than a year old on how to do that. But the principles would apply in the kidney just as they would apply in the liver or the brain. And so that would be useful. The second was for Mini's question around paraffin blocks. I think today, to the best of my knowledge, there isn't yet a good approach that works for single cell or single nucleus RNA-seq in FFPE material, but I do believe that multiple groups work on trying to make a technique like that work. Whether it would be successful or not is an interesting question. Joshua might want to comment on that because it's a combination of having RNA that is both in low quantity and in low quality. So even if you can isolate the entities, and even if the entities are sufficiently intact so that materials doesn't leak out of them, which are two ifs, after that you still need to have enough something out there to actually recover by sequencing. I think that's this trifecta of problems is why this is difficult. However, FFPE material can be analyzed by a lot of spatial multiplex methods, in particular those where you stain for multiple antibodies. And so even in those scenarios where you can't make the first atlas collection of profiles, say, directly from this material, you could then generalize your findings, both for testing them and for assessing them in the context of human genetics and other phenotypes and so on, by using the original information in order to define the right signature in order to measure in situ. And after you measure in situ, you can computationally basically port over the rest of the profile. That's something that seems to work quite robustly in other systems. I would believe would work for the kidney as well.
I just wanted to highlight that why don't we finish that thought and then, sorry, Josh.
I think you eventually you probably will be able to find a protocol for FFPE, but I would say you're making an atlas. Why not work with the best samples? Like if there's some particular sample that you can only get in FFPE, which I don't know anything about why that would be, maybe that would be like your secondary after you already have a good atlas so you could fill in. I know there's a method for neurons where you do something called patch-seq, you do electrophysiology, and then you pull out the contents, and then and this isn't what I'm saying to do, but the data are lower quality, and usually you overlay them on a higher quality data set, and then you can understand where they fit in. And so maybe that'll be able to be done. I think with even though the RNA is short, is small, it still should be fine for a 3' end method. I think that might still work. I think there was one other thing I wanted to bring up after that with Jimmy's here. I think the question is like, okay, are you going to do some sort of multiplexing? If you are going to do multiplexing, how are you going to do it? And some of these things require you to do, you might want to do a protease treatment to dissociate your cells that may have an impact on how you use antibodies later on for hashing. I think that's something that's just coming out. I think it's something you want to think about your protocols in the sense of prospective, what future looking, how will they fit into what's coming? Because I think most people are going to go that direction.
So I think maybe even before sort of getting into details of the different multiplexing methods, I guess a question for the community is if you are going to lose something by freezing down the cells, but you can do a lot more people at the same time, at what point are you willing to make that choice, right? Or you can do a few fresh, and maybe you determine from these previous sort of pilot data that fresh is still the best. Are you willing to take a little bit of a loss so that you could do better study design? And let's say that that's predictable, right? That we know certain cell type proportions will go down if we freeze the cells.
Yeah, why don't do both? So we're talking about proportions that we are getting a huge detailed description of the microenvironment, but still we are limited with thousands of cells, and we have sometimes with nephrectomies millions of cells. So first, it's important to know where you're taking the sample from. And it's good to also make annotations of the anatomy. So the spatial is very helpful. Also about SOPs that we hardly mention. It's really good to be in touch, and we can demonstrate that. Good to be in touch with the clinical team and get insights about the patients, about the sample, about the way that the sample was accrued, and so on. Yeah, I would say that with nephrectomies, you can also sample from few places. So then you can either compare between patients and within the patient. That's very useful, very informative.
So, and back to Joshua's point, a question earlier. So if you are trying to do different people, then I think one of the advantages of just leveraging natural genetic variation is that you have fewer concerns like what proteases are you going to use in digestion. Obviously, there could be allergenic effects, things like that. So you want to do these experiments rather quickly, which is what we do in my lab. And we don't quite understand exactly what happens when you wait and you have cells from different people sitting around. But yes, I would say that this is sort of a pitch for using the genetic variation. Of course, you can also do single cell attack-seq as well.
I think one more.
Yeah, so just as the outsider, as a lung person. So this is fascinating. This is exactly what we're dealing with, which is really cool. Could you introduce yourself? Naftali Kominsky from Yale. So one of the things, I actually think that numbers are really important. And I think if there's one thing we learned is, so doing the compromise, as long as you can have sets of perfect, inclusive, small set of really perfect samples, the rest you go for imperfection, but increase your numbers. So that's where we chose actually to use a data set of lung soups from both two locations, from Brigham and Women's and from Leuven in Belgium, because they existed. And they had already 100 samples. And we could perfectly identify what's missing there. But I think you have to have both, and that's the point that, and also multiple samples from the same patients are critically important.
I think one question which I also want to pose to the audience, nephrologists think about the kidney as a nephron, and we map our cells along the nephrons. We know that nephrons are different depending where they live. What effort do we have to systematically sample the organ above and beyond the simple nephron schema? Do we need the left and the right kidney? Do we need upper lower pole? Do we need to go through the different nephron segments depending where they are located? Their tubular cellular structure will be quite different. Opinions?
One more thing that I would like to raise is the issue of proximal tubular cells. So it just turns out that there are an awful lot of proximal tubular cells in the kidney, and if the effort is a cell typist effort to find the rare cell, one would have to think about ways of depleting them, because really the overwhelming cell type in the kidney is that, and that really makes it very frustrating. I mean, our effort wasn't really about finding novel cell types in the kidney, but if one wanted to go down that route, I think there has to be a way of excluding proximal tubular cells in the experimental setup.
Yeah, I think that's a great point. 70% of your sample are proximal tubules, so at a certain point you're really just wasting all your money on these cells, and there's no way to deplete proximal tubule with single nucleus RNA-seq. So that's a limitation of the nuclear protocols. To Matthias's point about the regions of the kidney, we've gotten one discarded whole kidney that was in good shape. We've gotten a lot that were not, as you pointed out. And we sampled it a number of different places, and the cell clusters are completely different if you're in the deep papilla or in the upper papilla. So I think this issue of if we only have biopsy material or if we only have nephrectomy material and we don't know where the tumor mass is going to be, it's going to be hard to get a comprehensive map without having access to a whole kidney and then really systematically taking pieces from the whole, from all areas of interest.
It's also not undoable with refractory specimens. I mean, it depends a little bit on your relationship with the pathologist, and it depends on the size of the tumor. But if you're there with the pathologist, you can take a photograph, and then you can say, can you sample all these, a bit of cortex, a bit of medulla, a bit of pelvis? Can I get 3 different regions, please? And once you added a bit of fat, a bit of blood vessel, a bit of adrenal, one can actually quite comprehensively sample a refractory specimen, and the only limitation that being the genuine clinical need for the pathologist to have to sample that particular area. But it sort of is doable, but it's quite intense. It does require going into the surgery, picking up the specimen, and sitting there with the pathologist looking at it together.
This has been something that we have been interested in, and in fact, we're lucky to have such a relationship with a pathologist, but it is very labor intensive. But we have developed a standardized, at least within our group, and maybe this is something to think about in terms of the community, kind of a reference of a kidney and then the location and a photograph and the location with like a drawing essentially of where the specimen came from, so that there's an approximate landmark or reference point as to where, are we in the cortex, are we somewhere in the medulla from a nephrectomy? I think that's entirely possible. Or even in the pelvis.
Yeah. So pediatrics is, in the UK is way ahead of that. So we've got a national app, as it were. And when Wilms tumor specimens are resected, pathologists can use this app or have to use this app and indicate where they've taken the samples from. They have to take a picture and they have to sample in a sort of predefined, rigorous way. That's not coming from an HCA side of thing. That comes from a pediatric oncology sort of end of things. And one could imagine that actually also thinking about the data portal, if you wanted to go down that route, one could have such an app for the human cell atlas kidney effort and then actually indicate this is where I took my biopsy from.
Do you think that should be part of the metadata that goes into these data sets?
That would be wonderful, because the truth is that data is only as good as its metadata. V1 just generating a few cells you can do without complicated metadata, but for the next level of insight we need that very detailed metadata.
How plausible do you think it is to get precise coordinates or information about where the biopsy was taken?
So in consonance with the idea of having a mesoscale map, there are a number of models spatially that have been proposed recognizing their reproducible fiduciary points anatomically within the kidney that can be used for a self-similar scaling model. So as long as you have an ontology which creates a central reference point, and you have some level of granularity in acquiring when you get the biopsy or sample where it's come from, it is possible to put as an annotation to the various samples the general location. And so there's different length scales. There's physical anatomic location, but there's also recognizing there's a depth going from capsular surface and cortex to medullary areas, and whether it's a medullary ray or more in the pelvis. Again, these may not always be known, but if you have at least a metadata framework, if it is known, you can add to it. So that just enriches the granularity. And the first step of being able to do that is at least create the framework so that the data is available. It can be stored at the time that the sample's acquired. Because as time passes, as you know, getting all the information after the fact becomes difficult, if not impossible.
Yeah.
So you want to have that framework in place ahead of time. And we haven't, like for example with KPMP, we have so many ontologies now, at least it's in the discussion in terms of the mesoscale. So I would just, in summary, I'd say you want to include the mesoscale as part of potentially the data capture.
Great. I mean, it sounds like we need to, on the ontology side, we need to be better aligned. I mean, we're using just Uberon at the moment, and I don't know what granularity Uberon has for kidney, internal kidney structure.
Actually, that's where we had a meeting with leading ontologists in August at the ramping up phase of defining metadata and ontologies needed. And I think a white paper is currently in generation, hopefully be shareable in a few weeks with this community, and then would love to get input. How do we connect? Which ontologies need to be built up further? There was a lot of enthusiasm from the ontologists present in the meeting to help as we engage with our domain knowledge to build that up. So I think this will be a critical aspect, and would love to hear, I think, with the other organ domains, there will be a lot of shared burden which could be lowered by putting on more shoulders here.
Yes. Yeah, no, I mean, from a HCA metadata perspective, we have a variety of JSON schemas, but we can totally have organ-specific schemas, so that there can be, because obviously what you need for a kidney will not be the same as what you need for a heart or a liver or whatever.
Laura, let me connect you with Oliver He and Sean Mooney, who are leading these efforts with, a couple of FTEs behind them to hopefully align things.
Yeah, we should get connected then.
So I think there was a theme. I think we're naturally moving to some of the computational considerations, and what I heard is that there's it sounds like there's this need for spatial information, and we can generate single cell or single nuclei information. I guess the question for the community is everyone okay with just mapping your single cell, single nuclei data onto a spatial map that's generated by fish or merfish or something like that? Or is it actually critically important that we develop assays where we can get both types of information in one go?
I think that, at least from our perspective, we're very keen on actually getting like real granularity at the single cell level with the spatial information. I think it's a very challenging question, but it is something that, we are actively interested in pursuing. I think for a highly architecturally complex and ordered structure like the kidney or like the brain, I think that these are worthwhile efforts to try to see what we could do. And I think, we can take technology from other tissues and see how it might apply to the kidney, and that's something that I'm personally very interested in thinking about further.
And coming back to Josh's comment, I think the beauty is if we can identify relevant defining transcript set for a cellular cluster, then with the FFPE technologies quickly emerging, we can go in high quality FFPE tissue, which will allow us to reach significantly larger segments to see how these signals are mapping, including, the questions about, which we had earlier, lifespan, ethnicity, environmental exposures, and how they impact the health state.
So staying on this spatial mapping theme, I think there are many different great methods out there right now, and what you really want to get out of them depends on how much multiplexing you would like to build in, how many different cell types you would like to detect, what is the kind of morphology you're looking at, are thin sections okay, or would you like to look at kidney volumes? So first off, is there any insight from existing data sets on the number of different cell types one would like to assay in the kidney, and the minimum set of markers one could use to distinguish them in situ?
I mean, with regards to number of cell types, it depends on depends on how much you want to subcluster and cluster and cluster and so forth. We just stuck to the canonical cell types, and off the top of my head, I don't know, in the developing kidney there were maybe, 10, 15, that sort of thing. Maybe in the mature kidney 20, 30, the markers that are already known in the literature are actually very good in differentiating one cell type from another. So I would have thought that in a combinatorial way, I would be surprised if you needed more than, let's say, 50 or 100 genes to actually provide some resolution. I think the question that we have here is a twofold about the spatial. There's the generating the reference atlas, for which I don't think it's necessarily a kidney-specific question. One just needs to think about at what point of spatial resolution do we say we have got the atlas. That's one question. And I think the other question is about what you were just alluding to, is the FFPE tissue when we want to go into different, biological states or disease states, and then whatever method one uses is a little bit determined by what tissue is available, but then also what it actually is that one wants to validate. And I think those are sort of two separate efforts. And for the atlasing effort, I do not know what the correct answer is of what spatial method one has to use, and at what point we say we have now got the atlas, and I think that's open to debate, and that's probably a question that is beyond this forum in a way.
Yeah. Well, I think it would be interesting to sort of detect areas of the kidney that are, say, repetitive nephron units that can be sampled at a different spatial frequency than, say, some of the more morphologically complex areas. So, you don't need like an atlas at the resolution of the brain one, perhaps.
So, related to the fact you have multiple cell types, we mentioned earlier the mesoscale, but that's way beyond the length scale of 1 or 2 or 5 cells. I wonder if it's appropriate to describe or capture the local milieu. In other words, we know, for example, that depending on what adjacencies are for a given cell, we have gap junctions, we have tight junctions. We know that cells intercommunicate with each other.
is there a need of having all the combinatorics once we've identified the cell types to say, okay, for this cell with this atlas annotations, these were the surrounding cells? And
I think that's what you're saying. That's really key. The kidney is not the epithelium. The kidney is the epithelium and the interaction with the vascular endothelium at the very least. And whatever spatial effort there is has to sort of capture that communication between those fundamental cell types. You know,
and that's more independent of where you are exactly in the kidney. I mean, it might have some bearing, but it's more based on the local microenvironment, right? Which are the exact adjacencies of the given cell under interrogation.
It is true, but I would have also thought that the perfusion of cortex and medulla are quite different, and it must make a difference whether you're tubal
absolutely,
up there or
but
down there.
I'm just saying not to discount the local microenvironment.
No, absolutely.
What would be resolution for you for mesoscale?
So that would be, if you're thinking about like cell-cell interactions, it would be at the cell length scale. So for this a priori cell, what were the adjacent cells? Maybe not know all of them, but some of them. What were the types of cells that were immediately in physical contact with this cell, if available?
So I'll comment actually on both. The first on the repetitive nature, and I am not a kidney expert. I actually know nothing about the kidney. But I was in the gut session yesterday. I'm also not a gut expert. That's also a repetitive structure, right? So my point is the following. I don't think we actually know. And so the question is, what sampling strategy of these repetitive structures would give you a definitive answer on whether they're just repeats and you can do a subset of them and you're done, or at what point they are not actually repetitive, there is a gradient, there is a something, there is a location that matters at that scale. I don't think anyone knows, right? I think people know at the level at which they looked in the past. But they might not know whether, maybe the local neighborhoods we have a little different, and maybe it's all exactly the same. I'm not actually putting a bet in one pot or the other. I don't know.
And then another challenge is that we are talking transcripts, and we know that, for example, the metabolite characteristics of along the nephron from cortex to medulla are a lot more dynamic than transcriptional. So to what extent do we want at least to be proactive that the tissue we are procuring for our transcriptional assays also amendable to metabolomic profiling, as you know, the technologies are getting down certainly to the mesoscale level and hopefully to the single cell level in the near future.
In systems where there is a gradient, there will be differences. But whether there is or there isn't, I don't know, and whether it's along one axis, another axis.
So in the kidney,
and how but
we already a priori know that
you know that it exists.
That there are significant metabolite gradients.
And the repeating units will still have some features that would repeat likely. So parsing that out, I think, is one of the things you want to get a definitive answer to.
So
on the other comment that was made about the local neighborhoods, at least in our experience from looking at other systems, besides we have experience from the brain too, but let's put the brain aside. Okay. But other epithelial tissues is that you have multiple length scales at which these phenomena are correct. And so there could be pairs of cells that like being partners of each other, and you would find them in many different neighborhoods, and they like to partner. And then you could have bigger neighborhoods of cells, kind of their combination together is repeatable. And then you can have even longer length scales. And these things also trace with what the histology looks like. So finding those, I would imagine, again, would be a very interesting question, and learning the right sampling strategy, not to overdo things that are just repetitive, but to get enough of them so that you actually trust that they're a thing, is one of the challenges.
So I think this is almost a computational challenge, because I think we've spent a lot of time as a community thinking about how many cells we need to sequence, or at least developing a framework for grasping this. But we don't really have this. We haven't thought about it as much.
Spatial power analysis,
I like that, spatial power analysis.
And I think what is critical is to use information like the one Matthias just conveyed. So you're not just sitting there, with your algorithm and entirely data-driven, which is perfectly fine, but it's also okay to use the fact that they actually already know something, right? It's we don't necessarily have to rediscover from scratch everything that's already known in biology. We could leverage that.
And
Bayesian spatial analysis, power analysis, like that.
You even can build your discovery assays to hold some of these truths out and then see if you are recovering and fine-tune your parameters that way.
Is this an opportunity to also, oh, sorry, Josh.
I have just another more general question, which is, how does the kidney project, or all these different organs, they all have interactions with some of the other ones, so the nervous system, the immune system, how do you decide where you're going to leave off and the other project will take on, or how do you integrate with them?
This is interesting because we were in the immune one yesterday, and from the immune perspective, it's easy. Just give us all the immune cells, and we can. This is actually one way of constructing sort of a separate immune cell atlas of the tissue, for example. But the nervous system, I'll let the kidney specialist.
This is where I think Paul again can comment from the AMP experience, where there's a focus on tissue-resident immune cell.
Yeah, so from the AMP, what we try to do is isolate all the immune cells. And if we were to just use lupus nephritis, which is a severe complication of systemic lupus, as our example, kind of a guiding principle, we find that there's roughly about 21 different types of immune cells. And there's 3 major categories, and they can all be subclustered from there. But I think it raises some important questions. Primarily, what types of modalities will we use for sequencing? For example, I defer to my nuclear sequencing colleagues on this, but the ability to actually get good sequencing information from immune cells using nuke-seq, I hear is limited. And I think that that's going to pose some challenges if we are very interested in understanding what the landscape of just a normal, healthy immune cell system looks like. And this is a key comparison that we need for these other consortia. I would argue for everything, because I think the immune system is going to be driving much of the chronic changes that we see in these systems. And so understanding what that baseline is going to be very important.
Can we think about this maybe in phases? As a first reference, maybe we don't do the best job of sampling immune cells in the kidney, but if we know that there are certain cell types that require special considerations, that we actually use more biased sampling strategies. And I think this came up earlier for either not to sample abundant cells even in the kidney, or to specifically design strategy for sampling immune cells. Is that a viable option for the community? Yeah, not as version 1.0, but like maybe a 2.0 version of the atlas?
Right, I think so. I think early on, I think technologically we might just be limited to that. I think many of us in this room would aspire to have everything come from the same sample and then go map it back, and that's really the gold standard that we're all striving to. But technologically speaking, I think what you're proposing will probably be the path that will be most viable.
I just wanted to add to that I think as we're looking at the normal kidney cell atlas, we're not looking at, let's say, lupus or transplant rejection, where of course you can study a lot of immune cells. I think if you look at the spectrum of age, in the elderly, you're actually going to find that tubular dropout and some tubular sclerosis, you're actually going to find that there's going to be infiltration of immune cells. So you may find that the impact of looking at some of the immune cells in these environments will also be present in the normal kidney as you look at age.
I just wanted to follow up on the immune cell question with nuclear, because I've had several people say, oh, nucleus is bad for immune cells, and we've not had that experience, and so I don't know where this is coming from, and I'd be curious if people have really looked at this. We find 3,500 unique genes per nuclear leukocyte, and my question would be whether actually the cell dissociation actually overrepresents leukocytes, because they don't have coherent tight junctions, and they're freely mobile, and they're much easier to release. And that with nucleus, maybe you don't see as many, but that might be a better reflection of reality. I don't know.
I think, Ben, you're right. I do wonder.
Dissociation tends to favor immune cells. Yeah, we know that.
Yeah,
And,
and that's been our experience, and I think actually that it might not be true about the nuclear. But leaving that aside, I think the immune question in kidney is quite important. It's a shame that Mena Clutworthy isn't here, who's obviously very interested in the immune environment of the kidneys. And in our experimental strategy, we also went for one thing, that after dissociation, we CD45 enriched, and thereby trying to capture the immune environment of the kidney as well. And I think that's a worthwhile parallel effort. I don't think there has to be a succession of you do this first and that. You just sort of go for what biologically is your interest.
One of the comments online is that one of the mics is not working. Do we know? I don't want to leave any. Oh, this one. Okay, but we'll fix it. Okay, fair enough. Just making sure that we're responsive to the online comments. Yeah.
So maybe going back a little bit more to the computational side. So I heard that there is, these efforts already happening in disease, KPMP. If we're going to generate a reference cell atlas for what I imagine would be healthy kidney, how do you guys think about, or how important is the integration of the data? And what would you like to see from computational biologists in terms of tools that would help that cause?
I'll drag it back into the spatial analysis again. I think, you know, like a common coordinate framework, a maybe averaged, perhaps micro CT-based atlas of several whole kidneys would be a good start. I think, you know, a lot of the stuff that we're talking about where you want to integrate the spatial measurements with the transcriptomics, I think there are a couple of like workflows that could be adopted to actually extract both kinds of information from the same sample. And I think it's best if it's done at the hands of people who know both the pathology and they're invested in dissociation and transcriptomic analyses. So I think what I would ask is, you know, if we wanted to do this for a whole brain sample, we would use an MRI scan. With the kidney, does it make more sense to have like a micro CT or some sort of other coarse imaging modality for starters?
I just want to strongly second how important that is. If you
And
want organ level, if you want an organ level atlas, having those types of things, MRI, CTs, and so on, is going to be absolutely important, critical, as is the anatomical registration when the tissue is actually sampled out. And in the brain initiative, one of the things that I think has served them exceptionally well is this tight connection with the anatomists. Partly because in the brain, otherwise you can talk about anything. That's how everyone thinks. So it's been very ingrained into the process. But when you obtain today a small piece to do your single nucleus RNA-seq in the brain initiative, it is registered in extreme precision to the coordinates. You know exactly where you took it from, both in the physical sense, like in the actual specimen you were sampling, and then that sample is sectioned, and then the section itself goes into this separate analysis through the kind of an anatomy pipeline to tell you where it's coming from. So of course, they're ahead of the curve because their coordinate framework exists already. In the case of the kidney, it would have to be built. Yeah. But
yeah. I'm not sure it really has to be built. I think the problem is that there is a lack of engagement with the pathological community. So so if we had pathologists here,
it can be brought in if it exists.
Yes.
It can be learned computationally, and it would actually change dramatically by computational learning.
Yeah.
But the point is, you need at some point, even if you infer everything from scratch, you would have to test that your inference is correct. At which point you would need to know where things came from. You might hide from yourself the information at first.
And actually, there is a very active community of adopting imaging technologies to the kidney. There's a large European network who is focused on that, including a protocol biopsy arm to that. So there is a way to link that up, and you might represent the pathologists at least to some extent here.
Well, just one thing I can think of, that not sure if it's been done, but it could be done computationally with existing tools and methodologies, would be to obtain a whole organ micro CT or micro PET CT, if you're looking for functional aspects, and then upon harvest of the organ, if it's an explanted organ, carry out whole organ serial sections with representative sampling of those sections, where those sections could be through auto-registration processes mapped back to the radiographic domain exactly. And that wouldn't be planar data, it would be voxel-based data. Yeah. So after the fact, you could go through the organ in any orthogonal plane based on your interrogation mode. And additionally to that, there are other modalities that are emerging from the obtained tissue which are non-destructive, which have now 1 to 2 millimeters of interrogation, which could be added, like the light sheet microscopy and other optical coherence tomography techniques. So it's not that they're not available, they haven't been applied in common use yet clinically, but they are out there.
And I think that's critical for these types of organ level.
And that will come back to our SOPs, that we procure the tissue in ways who actually allows these parallel technologies going forward, and ideally not only at a given time frame, that material is procured so that as the additional technologies come online, can be utilized and can be linked back to the existing data sets. So building a biorepository who allows these comprehensive analysis to move forward, I think would be a very valuable effort.
And I'll just say something while I'm going to, oh, okay, you're taking the mic there. The other thing that I wanted to say that may be an advantage for the kidney that we're not harnessing well enough, not thinking as much about at the level of a CT or the whole tissue, but actually for every clinical case, so every nephrectomy that happens, every biopsy of the kidney that happens, we are, as a standard operating procedure, we take EM, electron micrographs of the kidney, which is very rare for like, I don't know of any other organ when that happens in a standard way. And so for every patient, we have that level of information. And, you know, of course, in clinical care, there's a focus on particular structures, but you could actually go back to those samples and section them through and obtain more volumetric information, which on occasion we have done for, you know, on the research side when we're trying to, you know, analyze, you know, the efficiency of a particular therapy or efficacy of a particular therapy in an animal model. So we could actually reconstruct a lot of that information in terms of cell-to-cell relationships from electron micrographs, which is something that I think we have not harnessed at all and could be very, very useful.
That's fantastic. And I think, you know, down the line, it will actually make measurements across different sites, as well as the comparison of different disease models, much more reproducible, because you have this common framework that you're mapping your data back to. And something like a micro CT, I think, is much more affordable than having an MRI scanner in every different site, right? But I think some of that requires like forethought. Like, you know, if you're collecting all your samples and they're all frozen, then, right, it's like you're ruling out some of these spatial imaging methods.
So for the first thing about the electron micrographs, I think, just to speak to Sam's point, it means a stronger partnership with pathologists. So I think maybe one obvious thing is to bring more renal pathologists into this community would potentially be an advantage as we move forward. So I just want to say that.
I also think that what they could do is, in the first instance, provide a very, very basic anatomical reference framework that we all adhere to. I mean, you know, Avi says we need a reference, we do, but let's just start very basic. If we could just agree on what is cortex, what is medulla, what is pelvis, that would be a starting point. And there's a little bit of a shame that I think we are all the wrong people to do that. We are not pathologists. We don't cut up kidneys day in, day out.
Right.
And they are the people who identify these regions, and they are the people who need to define what it is. A bit like the anatomists in the brain, we need that equivalent engagement in the kidney.
And one last thing to say then, just to throw out a challenge back at the idea of the micro CT or whatever other modality we would choose. From a sort of computational perspective, how many of these do you need on how many patients before you would have a reference? Any thoughts? I'm just throwing it out as a challenge too.
If you use the human anatomy as a reference. Yeah. It would be more than one, but obviously this is an incredibly resource-intensive exercise. So I would think between 2 and 3, maybe 4 at the most, but wouldn't want more than that, at least as an initial exercise to see what the added discovery potential is.
And what about lifespan? So like I don't know, fetal or pediatric versus...
Well, you'd certainly want to consider the different, like, you know, healthy adult, pediatric, fetal, if possible, from discarded tissues, and maybe in terms of the geriatric populations, what the senescence looks like within the organ.
I wonder a little bit whether we are overestimating the complexity of the kidney. I mean, it is, of course, a very complex organ, but at the end of the day, the fundamental functional unit is the nephron, which is a module. And if you look at kidneys, you know, it's got sort of, I don't know, 3 segments, 2, 3 segments on each side. And actually, if we perhaps said, if we just try to leave out all the nuances, all the complexities, and the lifespan and all of that, and just focus on, let's in the first instance produce an atlas of just one nephron and do that really well, that could be a starting point then for everything else.
I also
I would say, just to, this is harking back to something Sam said a little while ago. When you do agree on what the different sections of the kidney are, I'm afraid I can't repeat the words you used. We need to make sure that that information gets integrated into Uberon or whatever is the right ontology. So then there is a computational record of that agreement, as well as everybody knowing. Just so you know, there's no you know, in a year's time going, wait a minute, what did we decide? So.
Question maybe for the DCP. Is there an opportunity to engage the community currently when they deposit data? Could you click on, for example, a kidney and say this is where it came from? Would that be possible? It seems like that would make things easier to help to find a reference.
We would love to have that functionality. We are a long way from that functionality. At the moment, the way data is being exchanged is conversations and emailing spreadsheets between wranglers and the people submitting data. And probably we're not going to be massively far from that in a year's time, just because of the complexity of the system that we're trying to build. But certainly, we want to get to the point where that's possible.
So I just wanted to also ask, what about race? So we know that it has a huge impact in disease progression, in incidence of disease, as well as in the context of transplantation in recovery of injury. So I mean, as we talk about the normal cell atlas, should we be also thinking about stratification by race and inclusion of some of that?
Matthias, you want to comment on that?
I think really this is critical, and I think I would even go beyond race, but really also geography and environments. And it gets back to the question, what are good numbers? And numbers are not only absolute, but also relative. And again, in the immune session yesterday, we had a very good discussion that these different genetic and environmental exposures will teach us a lot what normal looks like and how these will perturb the normal state. And ensuring that we are sampling, and I think, as always, North America and Europe are very well represented, but the disease burden is global. And reaching out into Southeast Asia and Sub-Saharan Africa is doable. Several of the networks I'm involved with are doing that. So we have access to kidney tissue out of Africa if needed. So these are options to discuss if this is something of value to this community.
Any other comments? I guess, is it worth it to consider? Oh, we have something from.
This goes back a little bit. That perhaps EM could be used.
Oh, yeah. So there's a comment on the live stream that EM perhaps could be used for all tissues in the HCA, and I don't know. I mean, that's an interesting idea, but we certainly are very familiar with that in kidney. We do it clinically all the time. So maybe, yeah.
Just circling back on comment about ontologies, what could be used immediately to make the data granularity better. I wonder about educational tools to be used at the bedside. So for example, if you had a tool where a clinician or surgeon could point to the organ, or maybe have schematized diagrams, a little bit, insets, higher resolution, and that would generate the appropriate ontology classification. Then you'd have consistency on input.
I think that's a wonderful idea. Practically, we're away from that because you need three-dimensional representations. You need people to have tablets at the bedside and all of those sorts of things. What we do have now, and we are very keen to improve, is basically a sort of trained text string to ontology term mapping service. And so, if different clinicians use slightly different terms for the same pieces, we can hopefully. And it's called Zoomer. We can add this to the system so the right ontology term is picked in the right circumstances. But yeah, again, ultimately, something like that would be wonderful.
It gets back, I think Jules' point gets back to the question, atlas for whom? So who do we want to surface this atlas? And I think that will critically determine how do we expose the data we are generating.
For the world, right? As representative as possible of all human kidneys. Yeah. If that's the case, then I guess one question that gets to study design, maybe that's what Jamie is trying to get to as well, is, and maybe I'm a very numbers-oriented, concrete thinker here, but really, how many people does it take? Like, can we do that thought exercise? And do we need hundreds? Do we need a few dozens? What?
Well, I think it's input the modality
exactly.
is.
Exactly.
And I think this is what we're hearing. Yes.
That's
I mean, we can, so I think, I want to leave it to you guys because I think there's some serious considerations about sample acquisition and then challenges of getting spatial information. But this is a really good question. What do you want out of your atlas as sort of like version 1.0? How many samples are we going to do single nuclei sequencing? How many samples do you guys want for mesoscale spatial information using something like electron micrograph? And how many samples do you want really deep sort of spatial information? Because it's clear that that's going to be important.
One way to start this is to ask among the people here who've already done single cell and single nucleus, what's the inter-individual variability that they see, and what are the differences in cell type proportion? So maybe people in the audience here who've gone through that exercise among maybe 10 individuals can comment. And what is the notion of normal in relation to the other pathological features that come with it, metadata?
I'm going to take that question because I think it might be challenging for a community that they haven't done hundreds of people, right? And in one group. So I can give you some perspective from, for example, doing hundreds of people in immune cells in a very
sort of targeted way. There, from person to person, the variability is up to about 20 to 30% for the major cell types. Now, the repeatability of the same person in these assays is, that variability is less than 2%. So I think the technology is good, at least for single nuclei sequencing and single cell sequencing.
I think one aspect we have is that we have bulk data from several thousands of patients in the public domain, biopsy derived, so it will be primarily limited to cortex. But if there are parallel systems where information linking bulk to single cell data is available, then one can help with the bulk data to at least estimate what variability might be present in the single cell state.
I can just comment not from kidney. I don't know anything about the kidney, but two things. First of all, to Jimmy's comment, there are two levels of variability you might want to ask questions about. The first is compositional, so the proportions of cells, and the second is intrinsic, so the programs that the cells express, not the same thing. The second is, and one could be much more variable than the other and vice versa. The second is that from our experience in the colon, and that's actually a paper that's posted on the bioRxiv, so people can look at it and get some sense, our experience has been that in healthy individuals, the variation is quite low. And both for compositions and most definitely for programs, and there we had 2 samples taken from the same individual but at the same biopsy. So 2 separate biopsy bites but in the same session, not recalling a patient, and that was basically non-existent variation. That goes to the repeatable structure in the colons and supports Sam's hypothesis that there would be very little difference, but they're close to each other. They're not far away. I would say that gave us a lot of hope, and that was based on 10 healthy individuals that were scoped, all at MGH.
Yeah. As in the kidney, obviously, we have the challenge that depending on your hydration status and other functional state, the function of the nephron segments will be in very different activation
Absolutely,
states.
And it is true that in colonoscopies, people fast and cleanse before the procedure, which definitely reduces probably a certain source of variation.
And the same is true for if you do biopsy procurement, that patients are fasting. They are usually done in the morning, so you will have standardized for diurnal variations and other aspects. And it gets back, I think, also to a larger question. Do we want already use the data we are generating to infer physiological functionalities from? Yeah. And physiologists, I don't think, are in this room and usually are not very engaged in the discussions we have here. And renal physiology is a very rich field where a lot of molecular information already is integrated, and bringing individuals to this community with that expertise, I think, would be very helpful.
And at the same time, not to complicate things too much, I suppose, again, it comes back to what do we want from our first iteration of the atlas? And, don't we just need to kind of get a fundamental understanding of what the cells are and their relationships?
I can probably comment on the variability in the kidney, and actually also one comment on the immune cells. I do not think you can extrapolate from the peripheral blood count to a solid organ. The normal range of peripheral blood cells is humongous, and it varies from, from children to adults. And it's got sort of sex differences. I think that's not a valid comparison or a valid extrapolation to make. As for the variability in the kidney, so when we looked at, let's say, we had couple of patients where we had like a couple of cortical biopsies, a couple of medullary biopsies, and actually they're quite comparable, the cortex with the cortex, the medulla with the medulla, but one medulla from one patient is more closely related to the medulla from another patient than to the cortex, of course. I actually think within patients, the regional variability didn't seem that great within the few, within the limitations of just having had a few patients where we got multiple biopsies. And however, there obviously is a difference between cortex and medulla, which, however, is utterly unsurprising. So
that's composition, by the way. So if you're looking at cell type proportion, there's little variability.
Yes, okay. And the other variability that we saw, which was quite striking, was between adults and children. And I just wonder whether that comes down to the children's kidneys somehow not being as fibrotic or as sort of gooey as an adult's kidney, but I don't know what the correct answer for that is.
Yes, we've only done a handful of these also, but have noted some unexpected differences with gender. And I'd say the biggest source of variability is what you just said, Sam, and that is age. We see entirely new clusters that we cannot annotate and don't yet understand what they are, and we see more of them the older the patient is.
And this is concordant with the bulk data, that you see the first principal component is gender and the second principal component is age.
So, you know, as a human geneticist, I think I'm a big fan of having large samples. However, it doesn't always have to come from different individuals. It could come from longitudinal data, or it could come from individuals different ages. So, you know, the one thing is it is, I think, cost effective today to do hundreds of samples. So the question is, if you could do hundreds of samples with single nuclear sequencing, I don't know about the imaging as much. If you could have access to hundreds of samples, you heard from the gut and the lung as well, how would you want to split up those samples in terms of age and individual variation?
Just because it came up now repeatedly, right, also with the regarding the GTEx, is it actually worse in terms of standard operation procedures to kind of collect also bulk RNA-seq information, and if yes, how? And
can I put a plug and say absolutely yes, absolutely, collect the bulk, right? And the reason bulk, and in the case where you can sort cells, sorted populations and their bulks, and the reason for that is that especially the massively parallel methods, at least today, are using a pre-existing annotation in order to quantify, and the quality of your annotation matters, and the quality of the annotation is often not so great. So the bulk you want to collect is the bulk that you need, which is what we call annotation grade. Long reads, paired ends, this and that and the other, analyze, build an annotation, use this annotation in order to quantify the gene expression. And in addition, it's a quality control that you can see whether you can assemble back the correct bulk and deconvolve the bulk, or whether there is a residual there that looks like a real signal that might be a cell type, for example, that you're missing.
Yeah.
So, yes.
And I mean, you can refer to all these thousands of samples that are already there, right, which might actually also be not so unimportant. Yes. But then, I mean, after having talked so much, of course, about the single cell, then we should think about kind of how one would do it, and with methods, and whether we would need benchmarking there, or just do kind of the GTEx approach, or so how would we, because I think that came out of people's mind, right, because they're heavily focused on single cells, so nobody does bulk anymore, but I think it would be actually very useful in the atlas.
We can also do a power analysis, actually. If we have some single cell kidney data, we could go back to GTEx and try to do decomposition and deconvolution. Yeah. The former problem is easier, the latter is harder. And to get an estimate of the variability, at least computationally, to that can then perhaps inform of how we design the study for the first atlas.
And no, in our series, we actually have done that, and Paul and Raji in the group has compared single cell, single nuclear versus bulk, and also versus laser capture microdissection of nephron segments to see how those line up with the single cell data. So these are different spectrums of aggregates which should help to define the degrees of freedoms you're pulling out and your blind spots.
Just to the KPMP initiative, what have you seen in terms of bias towards a certain section of the population in terms of sample collection? Do you see more samples from older people or younger people, and how have you gone about?
So the protocol biopsies are to be initiated in the next few months, and the clinical sites for recruitment have been selected so that we have a broad representation of the population in North America, particularly of segments of the populations highest at risk, meaning minorities. And our patient representation in KPMP purposefully is reflecting that as well, so that we can assure we are reaching into the minorities who are carrying the disease burden here.
First, I wanted to bring up a question from the live stream. Do you think there should be a consensus based on how many minimum cells as a percentage of total cell sequence is required to annotate a cell type in a TISNI? And I ask this as Atlas is good, but annotating them should either be consensus based or flexible, so we can go and fix them later. This is someone from listening in. Any answers or comments on that? So we.
I don't think it's a cell number problem, because things either separate as a cluster or not. But obviously you can manipulate your resolution ad infinitum and fool yourself with new cell types. But the issue of annotation, I think, is a more fundamental one. And we spend hours and days arguing over what we call what, and it really, I mean, that was the greatest source of aggravation for me over the past few months. And it would be lovely as a community to be able to sit down and say we all agree on that this marker is for that cell type. There's That's a great thing from the NIH. There's a group there who did something on mouse mini bulk, I think. And they've provided, I saw sort of...
Mike Neber has a community effort ongoing.
Yeah.
He has spent the last decade or two in defining the cell spectrum of the kidney.
And that sort of cell typist should really be involved in all of this. So, you need the pathologist, but actually having a reference cell typist or a few people who consider themselves to be the reference cell typist would be greatly valuable and would everyone spare a lot of aggravation over the next few months as they annotate their cells.
Mm-hmm. And we can certainly, one idea for some of the other communities has been a Slack that allows people to participate from wherever they are. And that way, someone who has that kind of experience could be, putting in a document with their favorite list of marker genes for each of the segments of the kidney. And I think it can also be sort of a grassroots efforts where others contribute and even argue. We all go through this discussion about what are the best markers for each cell, but that may be a way to vet this as a community and have anyone from anywhere in the world participate in the discussion.
What's interesting in the immunology discussion yesterday is the same topic came up, and the consensus from that group emerging was, define cell clusters by their transcript sets. Don't spend decades arguing how you label that, at least initially, until further functional studies can really tag them appropriately.
I just wanted to actually just ask the community how we should do this, because each of us has 2 kidneys and the huge redundancy in the function of that organ. Unlike other, I think, organs, the kidney can mask a lot of injuries. So if you're actually going to take only very few number of samples per group or per age or per gender, and we're going to develop the first atlas, I would also say that as a group we should define what is normal so that we actually have other parameters that actually get logged in. And we should decide as a group, is it just going to be the creatinine of that patient? Are we going to need some kind of imaging? Are we going to need some kind of biopsy, light microscopy, electron microscopy that goes with it, that actually classifies that because we have small sample numbers, that we're absolutely selecting something that we think is normal?
I think that's definitely part of all the metadata and everything else that we're talking about here. So absolutely we have to have that information. And there should be no evidence of kidney abnormalities at all in those who we define as normal, for sure.
I actually want to go to the point I originally went to make and forgot to make when I raised my hand last time. Ended up talking about annotations. Jimmy raised the point of, with say, nuclei sequencing, you could multiplex a lot of individuals, you could get to large numbers. And then kind of said, I don't know about the other methods. And I think it would be useful for this group to think about what would you want to do in a large number of individuals, and what would you just want to do in a smaller number of individuals, but very deeply? Because there's a way of parsing this out that would optimize the answer that you would get. Some methods would not be good for large numbers of individuals. Some specimens would be a once in a lifetime opportunity, and you would want to go very deep in them. I think you would know better where to position those things. There are definitely assays that work really well at the large scale, and there are definitely assays that not yet, at least.
Yeah, I think obviously all the sequencing-based assays, it comes down to cost in many cases and access to sample. And so I think I sort of sketched out what I think is possible. But for you guys, I think it's really important to figure out what are the things that you want to do to a sample very deeply. And what I heard was basically some spatial analysis, but I would love to hear more about what kind of other assays you want deeply, including sequencing. You could say that, look, for a few samples, right?
You can do something for a lot of people. Does not necessarily mean that's what they want the most.
Yeah.
And maybe this also is a segue into, because we have to address that final kind of piece of our discussion today also is, as we think about the sample and how we're going to interrogate it, also the validation piece comes into this as well. Some of the validation may occur in those same human samples or adjacent or other samples that one would collect from the same patient at the time of the interaction with that patient and obtaining that sample. I'm thinking, for example, contemporaneous blood draw would be perhaps informative of the immune cell population in the kidney versus not. Other ways that you might want to interrogate that tissue to validate the markers that we will all be agreeing upon, or any new states that will be defined as we do sequencing of those samples. So maybe this is an opportunity to think about exactly what Aviv just said. which modality at which depth, and that would define what tissue you need as well, or what sample you need to be obtaining.
I think that's where orthogonal data sets are critical, particularly proteomic and metabolomic studies, because we will infer functionalities in these domains from the transcriptional data, and having the next layer in the multimodal system available to see where they track and how they track. And again, as we discussed yesterday in other organ system, metabolomics, for example, can be significantly more sensitive for different functional states than the transcriptional state might allow us to do.
And that, of course, creates a computational integration problem as well.
Opportunity, not problem.
Layer.
Opportunity.
Okay, very good. A challenge that we can address or meet.
So do you are you proposing to actually run some of these assays on, you know, I guess some of the spatial analysis is basically a protein measurement, right? So are we talking about running these on a few samples really deeply at the single cell level?
I would strongly recommend that we have a set of samples where we have high integrity, and be also wary that particularly metabolomic data sets are easily influenced even by very transient challenges to the procurement process. But if you get a sample set where you can layer these deeply on top of each other, then they will be informative, and you should be able, at least in several instances, then to infer in those samples where you're more shallow on a transcriptional level where these states are.
If we would think of, oh, sorry, Josh, go ahead.
A quick point on the proteomics. I think the site-seq method, you could already begin to incorporate, 40 markers into your RNA-seq, and you don't have to wait until validation.
So of course, the challenge there is that it has to work on single cells, right? So we have to make that compromise, but I think it's worth doing. And I'm going to write it's up to 250 markers.
Sorry, the other thing that I wanted to say is if we start to think a little bit outside the box beyond, and maybe I'm jumping too far ahead, but what about the possibility of orthogonally doing validation work in other systems? Is that something that we want to consider as a community? Our group is doing work in organoids. They are, pluripotent stem cell derived structures. And, there's an ongoing effort in the overall community to assess how useful they are, how reproducible, how they recapitulate development, or how useful they might be for disease. And potentially one could ask, are they useful in any of the validation work that we're doing for the Human Cell Atlas? Or mouse, which came up in the immune discussion yesterday, and there were, I think, fair to say, pros and cons in that assessment. But just to open it up to what other validation could we do? Should we stay with human and just think more creatively about more deeply phenotyping or characterizing the donors of our material from which we're doing our atlasing? Just putting it out there as a...
So, as a novice to kidney biology, are there sort of assays that you could do in vitro that could be multiplexed, or where you do it in high throughput? Is that at all possible?
I think there are a couple of aspects here. One is, which we touched earlier, crossing species, yes, but maybe not in scope for HCA, but actually going across organs in the same individual. And one aspect is certainly reaching through the kidney one way or the other. One cuts through skin, fat, muscle, and these would be obvious tissue sources which could be acquired as a sample is acquired from a biopsy process to a surgical procedure, and that could transmit to defining the state of the vascular in different organ system, defining the immune state in the same individual with the similar experimental constraints how to procure that material.
Argument could be made, just to counter to that, that you could additionally, there will be atlasing of those tissues as well. So you can you're suggesting from the same individual so that you have that data.
Obviously, if you have a limited number of individuals where you can identify where in an individual these systems are linked, then you can go across individuals going forward by triangulating the associations in the overall human cell atlas.
Yeah, argument could be made for the fact that perhaps then we should be busy thinking about how to incorporate data that are obtained on the clinical level on these patients who are or not patients, but donors, people who may be just interacting with, let's say their primary care doctor, and you could document whether they have high blood pressure, other things that could be driving the health of their vasculature, for example, which would ultimately affect, kidney states as well. But is that too complex? Is that too early to be thinking about that? I think Aishwarya has a comment.
I just wanted to add that the GTEx consortium has done precisely this in postmortem samples. They've collected multiple tissues from the same individual postmortem, and they have guidelines and systems in place to take into account clinical information and other metadata that's available. I think they're also currently looking into how to expand this further for single cell and single nucleus type work.
The only problem with postmortem sample is that the integrity of the kidney is probably the lowest in the entire collection, and thereby I think we are probably limited with that concept. Even warm autopsies, which are done in some studies with patients with defined disease stage, so consistently low quality of the kidney material.
I think Aishwarya is basically saying that we could learn from them so that we could layer the clinical data onto whatever new data we're collecting.
I would just make a plug for model organisms when we look beyond the Atlas 1.0 and begin focusing on disease. Obviously, with human samples, take AKI, which is of interest to the KPMP, you only have one time point. It's the time point when you did the biopsy. We don't do multiple biopsies in AKI ever. And with the model organism, of course, you can do ischemia reperfusion. You can get a baseline. You can get 4 hours, 12, 24. All the way to 6 months. And I think that kind of data set, even though it's mouse, will help us actually interpret AKI in a human and what are these clusters and what's the progression of injury and repair.
Perhaps, if I may add, so with regards to validation, I think at the end of the day, it comes down to what it is that we want to validate, and then one just uses an orthogonal method and an independent cohort in one way or the other. Now, in terms of the Atlas validation, I wonder whether your system, Minnie, might be quite powerful there, that if you can take biopsies from live donors, that you do your initial Atlas on the one side and then take the contralateral kidney to validate what you think you thought in one kidney.
Yeah, you could do that, and in fact, it could be interesting because these kidneys will go into different recipients. Yeah. So you could actually take a biopsy from each kidney, and then you could monitor its progression in the genotype of a different individual and actually look at progression over time. And there we get serial sampling in these patients. So I think that could be very interesting.
I mean, I think for organoids, I think it's important how the HCA in the end will see that. But I guess one would need the human cell atlas kidney first, and then one can validate organoids based on the physiological real data, right? And I think only this direction really makes sense. For the mouse, I mean, there will anyway be mouse atlases, right? I mean, there's just no doubt because one just needs them. I think where the biggest impact could be if you really want to validate some methods in order to see where are the biases and where are the differences, which genes do I get and do not get, then I think the mouse is really probably the best option everybody would have, right? Because that's just the easiest thing, to take a black 6 and then validate that on that. And that will not be, that's very unlikely to be fundamentally different from humans, right?
Well,
so, I mean,
there's some considerations. I mean, actually, from the perspective of the kidney or the cardiovascular system, it appears that rats are better than mice because the mouse doesn't really have the kind of blood pressure dynamics that a human has. Rat seems to be closer to that. But then you're getting into how representative is one of the other, and the question is if you're looking at specific, validation of specific genes in locations within the unit called the nephron, then maybe any of the above is okay. The mouse would be okay, the rat would be okay. Yeah. I think it's so we're not really looking at disease states. We're sort of looking at normal or physiology and how to validate that. But maybe at the end, it's really coming down to an independent human cohort with an orthogonal methodology that can really help us. Could that be codex? Could that be, some other way of getting at, it depends on what level one needs to validate something at, I think. But yeah, please.
So Naftali Kaminsky, one of the things that came up yesterday in some of our discussions was the potential of using a reference from a large species. So actually, in my lab's experience, pig was really good. When you get the lung really as the animal is, is being sacrificed, so it's really live. Relatively similar physiology. They're not standing, but apart from that, so the question is, it's not a big effort. It's just adding a few lungs, and you have this really perfect single-cell atlas in some ways. So the question whether the way you could feed in is whatever animal you feel is the closest to human, not be stuck on rodents. Right? And I would guess that it's probably the pig. And with the emergence of transgenic pigs, you can imagine actually validation studies in the future.
So just sort of actually, in an ideal world, you use the best mimic for model organism for what you're doing. It's worth bearing in mind the genome quality, the annotation quality, all of these things for things that aren't human and mouse starts to diverge. And so that brings in additional complexity. Obviously, they're getting better all the time with PatBio and with full-length cDNAs from and things like that. But there is a compromise when you go to species that are further away from human and mouse.
But one aspect is, we don't have to speculate. We can look at data. In the sense that as data sets are available from these species across their kidney state in bulk, you can already infer which principal transcriptional landscape is present and which is not, and where they map and where they don't map. And that might help to guide the selection, which of these organ systems are most informative. And I want to come back to Sam's comment very early on with the organ on a rig, which I think is a unique feature. If you have a defined physiological state, which you want to manipulate, again, like the immune discussion yesterday, then this might be a unique setting where physiological states can be exposed and then transcriptional responses to those states assessed, to get back also to the question, cell state versus cell type, what's plasticity present in our data sets, and that might be a neat additional way to consider.
Any further comments on validation efforts or things that we can learn from other organ systems, perhaps, or something we are missing or didn't talk about today? I think we also tried to see if there's comments online and talked about them. Welcome.
Just maybe wrapping up, because it's not clear. So if there would be a recommendation saying, okay, if you do your contribution to the atlas, of course, and you do your favorite nuclear RNA-seq or single-cell RNA-seq methodologies, then a recommendation could be, okay, also get a bulk sample, right? I mean, and use whatever it is, standard TrueSeq or what the heck. And then the other thing would be that one should definitely get a sample prepared at a similar location that then can be used for spatial analysis, right? And that's, I mean, that if that's possible just from the way you collect these tissues, and I guess it probably is, right? Then that's already a lot, right? And I'm not so sure whether, in addition to the spatial validation, which after all has probably the highest sensitivity and this additional information, I'm not quite sure whether one would really take the efforts to do a lot more, but rather this consistently.
Yeah, I think doing things consistently is obviously the way to go. And of course, the other point about getting a bulk is it helps you do some of these multiplexing experiments that we're talking about, so that the bulk RNA actually acts as a barcode. So when you do mix cells from different people and do single cell RNA or nuclear sequencing, you can call genotypes on that bulk. And a lot of people have been doing this, and it's actually quite helpful. So one thing I think, maybe since we only have less than 10 minutes, is in the immune session, I think it was really helpful to think about what are the next steps as a community. So how do we come together? What will help us work together? And maybe, we have a few projects here. How do we coordinate these projects? As you mentioned, some of the spatial analysis is going to be very challenging, and maybe not every lab should be taking those on. So I just want to open that up to hear what you want.
So I just have a question before next steps, which is still kind of high level. In looking at all these top-level columns, thinking a little bit out of the box, I wonder if we've perhaps left one out, which is from the perspective of the end state, which is somebody, various use cases, interrogating the atlas. Is it germane to have a column of what are all the possible interrogation pathways or modalities going into all this primary data that we're going to curate? it seems to me that there may be some aspects of renal pathology and just kidney and the nephron that would engender a specific deep dive into if there are unique ways of interrogating that data that might not be covered by the other organ systems. And then maybe it's already done, but it seems to me that interrogation itself is the end all of having an atlas. What do you do with it? You look through it.
And do you have any specific thoughts or something that you've developed at KPMP that could be applicable here that
Well,
we're missing?
It crosses my mind since the kidney is actually a bilateral organ. For example, if you had, maybe the data is not even available in all cases, but information about the bilateral organ that could inform the present state. Like we know physiologically with hypertension from renal artery stenosis, contralateral, but damages both organs. Of course, we're talking about normal. I'm thinking also in parallel with disease status, but there could be larger physiologic states that inform the normal condition as well.
To me, I see that as the kind of the integration of like metadata and possibly clinical level data that could inform the interpretation of what you're seeing in terms of cell states. And I think that the idea would be that there would be an effort to actually capture some of that and maybe make it, put it into a format that you can interrogate or understand further. But it's an important challenge to
like,
think
for example, electron microscopy was mentioned.
Right.
So a spatial query that combines a spatial question in the EM length scale and the microscopic length scale. I think that might be unique.
Or the single cell scale.
Single cell, exactly.
Yeah.
Asking what will people actually do with this data? Like so I think this could go actually into the computation. I'm sure
it could be, but it's a little bit nuanced.
It is nuanced.
It's not the pipelines. It's actually the user experience in the front end.
Yes.
How you approach the data as an interrogation.
I would say that that's actually critically important for the computational part. It's not just going to be about the pipelining. Our ability to query this data set, build APIs, I think the DCP spends a lot of time thinking about that. And also enabling the rest of the computational community to build tools on top of that that might be more clinician-facing.
And this is clearly a question which transcends the kidney and is relevant, I think, for all organ systems, and where human cell atlas is a human and not an organ-specific atlas. And the only question is, are there unique features of the kidney which might not be integrated into the standard build of the human cell atlas?
Yeah, that makes sense.
And there, I think renal physiology, I think, is a key feature where the kidney can be very rich, and we can provide information to model, which in other organ systems might not be as obvious.
Sure. Oh, yeah.
Just to say, as Jimmy's mentioned, the DCP is very keen to know the sorts of queries people ultimately want to be able to do, because that helps us move towards it. We're not going to be able to support it right this instant, but if we know what they are, we can aim for it.
And Laura, we do a comprehensive outreach in KPMP that's already underway, and I can loop back to the group, and Becky Steck, which I think you already met, is the point person there, and we can bring some of the key lessons learned back, which might be helpful even beyond the kidney.
Yeah, totally. We should make sure that information goes to our user experience team, because they're really interested to know these things, so they can guide our design in the right way.
That's perfect, because it's a segue into exactly next steps. So that's one next step on the practical side of things. But are there other big picture next step items that we would like to address? One other thing that I took away, and also thinking about the online community interacting with us today, is clearly an ongoing forum for this kind of discussion. And I use Slack all the time, so maybe I'm very facile with that, but is that a way, as other communities have done, that we could continue some of this dialogue and maybe bring some new people into this community? We talked about Mark Nepper and other people who have done a lot of the kind of detailed cellular phenotyping and ID marker identification. So we could reach out and invite others to join the Slack in sort of a very open way. Other practical aspects, things that, yes?
Would there be, I mean, is there a worry that maybe some cross-site comparison would be of value and even possible? I mean, having one specimen and then the centers that are involved kind of do the same thing, and then one gets at least an idea about the variation introduced by subtleties of the different people? Is that something that would be realistic? I mean, that again, first back to the mouse, right? You could in principle do that with a model organ, because I guess if you have a transplant, that's not really, you cannot send it around. But I don't know, right? I mean, I think cross-site comparisons might eventually be an important thing, even, yeah, because that will be a practical thing. People will do their own assays and then will ask, I see differences.
See, Wolfgang,
can I explain that by just cross-site differences in principle?
You're making a very important point there. So I come from the cancer somatic mutation calling community. That's my background. And within the ICGC, the International Cancer Genome Consortium, what we used to do every year, someone extracts some DNA and then sends it to all the different participating centers. Everyone generates sequences and runs them through their algorithms, and then at the end of it, we compare. And I think that is the way to do it. So, the way to do it would be, Mini gives us, 25 different biopsies. You send it to everybody who wants to be involved in this, and then just, knock yourself out, generate yourselves, annotate them, and then we compare. And I think that comparison could be very powerful to teach us about technical issues, but also about the interpretation of the data. Because in a way, the annotation is the manual kind of mutation calling that we do on some level, and that needs to be subjected to some rigorous analysis and comparison across sites, absolutely. And that way, we can actually develop a gold standard. Otherwise, it'll just be, everyone does their own thing, and we don't know whether at the end of the day, that's valid. Yeah.
I'd like to comment on that. So we've done this in a couple of the consortia we're involved with, and the idea is to get the same patient and then send them to different sites and then have those sites do their whatever protocol and perhaps even do their own analyses. And what we find is that in general that there's a big difference in their technical variability, not surprising. So then it raises the question, it raises a few questions. If there's technical variability, then you're going to introduce tremendous batch effects, or possibly. And so then it raises the question, like, what are, is there one site that should be doing this? And then it comes down to funding issues. And so then it starts getting into kind of a tricky terrain to navigate.
No, you have to charge the variation, I think. I think that's really what you need, right? And there's no way that you would say you're the best and you do it.
No, that's right. But then do you want the batch variation between the technical sites? And what is that? There's going to be differences. Are they technical? Are they biological? Do you just take the things that overlap and call those biological? So I'm not.
But that's why you take one sample, right? I mean, in order to exclude biological variation.
That makes a lot of sense. And initially, I think that, that marries us to single nuclei as a method, but we could certainly obtain samples and ship them around and actually do that. And I think that could be a kind of a QC step as we develop Atlas 1.0. I think we have come to the end of our time, and I just wanted to thank each and every one of you for participating in what I think was a very productive discussion. Of course, it never ends, and it has to continue, and so I think we'll try to continue on the Slack channel and then through other avenues as well, and maintain, kind of active participation in the community. I think we will try to synthesize everything that we discussed today the best we can and try to present to the broader group, but please keep us true by jumping in and saying, hey, we also talked about that if we happen to miss something. And I think that we will try to develop some sort of one-page summary of what we talked about today and make it available to everybody. And in the past, the way that we've done it, and I favor that, is it could be a living document, like a Google Doc, where people can just jump in and add or edit or annotate in any additional ways, and so that we can keep it going. And if you have any other ideas at any time, please let any one of us know. So thank you so much for your participation today. Thanks.